Okay. So microorganisms and their benefits to the human beings. Okay. So in what ways are they beneficial? Now we know that there are two categories, right? One is the harmful microorganisms and other one is what? Beneficial. So in this chapter, we'll be seeing about what the beneficial microorganisms and how does it play a role in various formation of the products or in various processes in day-to-day life. Okay, so usually let's start with the categories one by one. That is the first one which we'll be seeing is the household category. Okay, so now as you can see over here, now these are what the types of microorganisms. Okay, so depending upon where they live, okay, what is their habitat or in which environment can they survive, according to them we have what several categories in microorganisms. Now basic things we know that in the microbes, what are the categories? It'll be the bacteria, fungus, then protozoa, virus, right? All these comes under what the classification system. So these examples which you can see over here is what related to on what habitat or in what type of climate can they survive. So the first one is what the thermophiles. Okay, so usually thermophiles means what they have the ability to live in the extreme hot conditions like the hot springs. Halophiles means what? Halo means salt loving they will be. So usually deep under the sea you will find them. Next you have is the psychrophiles. So psychrophiles means extreme cold conditions. Okay, that is their optimum temperature being what? 20 degrees to they can survive up to 0 degrees. Then we have the extremophiles. Okay, so extremophiles are the categories which in which we have what the alkaliphiles and acidophiles. So depending upon whether they can live in an acidic condition pH or a basic thing. Okay, so these are what the categories of the microorganisms depending upon on what temperature on what basis they can survive. Okay, then the next one which we have, just a second. Yeah, these are what the shapes of microorganisms. So basically we have what coccus, bacillus, vibrio, cocobacillus, spirillum and spirochete. So depending upon what type of microorganisms they are, whether they are a bacteria, whether they are virus. Okay, so depending upon they have what different shapes as well as sizes, okay, which may go to up to the nanometer. Then next what we have is not. Yeah. So this was the thing which I was telling you in the starting. Okay. So depending upon the nutrition requirement, these are the basic categories that we have. That is bacteria, fungi, protozoa, algae, helminthes, and viruses. Now, out of this, in this chapter, we'll be mainly dealing with the bacteria and fungi, which will be used in what different processes. So when we are culturing these bacteria or fungus, okay, whatever microbes are there, when we culture it in the lab in a sterilized condition, what happens is we use something which is called as what a nutrient media. So this is called as what the nutrient media. So this can be done in two ways. One is either in the form of a broth, which is liquid consistency, and over here which you can see is a solid nutrient media. Now what is basically a nutrient media? Okay, so now if you see there is like transparent yellowish or light brown color on the petri plates, right? And inside that you can see small dot like structures which have been grown. So basically the translucent or transparent thing which is there, that's your nutrient media. So here you can see the bottle over here. This is the nutrient agar powder into which we add different what do you say, extra nutrients, like it may be the nitrogen or phosphorus, whatever is required for that particular bacteria to grow. We add that into this agar powder. So agar basically what it does is it acts as a medium base. For example, you may know about the gelatin, right? So when we make jellies and all those stuff, what we use is the gelatin. So what does it do? When you mix it in the warm water, and once it's cooled, it sets out, right? So the same role is played by this nutrient agar, which acts as a base into which you have to add all the nutrients for the microorganisms to grow. So when you grow certain species, like for example, take any type, maybe it is an E. coli, okay, or here as shown over here is the salmonella, right, a Klebsiella. So all these are what different nutrient medias are required for them to grow. Okay, so this is basically the small dots which you see is what the growth of the microorganisms. Okay, so this is all about what the basic, what is the shape, the basic types of microorganisms and all those things. Then coming towards the next thing. This one, yeah. So as I told you that the first category that we are going to see is what? The household thing. So yeah. Okay. So this is the flow chart which shows what how yogurt is made. Okay, now there is just a slight difference between the curd and the yogurt. Okay, so but the whatever procedure to make it is almost the same. Okay, so when we say curd, now, curd is something which we make from the milk, right? So, household, like in your houses, when the curd is made, it's a very simple procedure. That is, whatever the first produced curd will be there, take a small spoon of that and put it into your warm milk, right? Then, once it sets, you get the curd. But this is not the case in case of the industry. Okay, there is a certain procedure or protocol that should be followed, and mostly whether you're making it at house or in the industry, there is an important microbe which plays a role in the curdling of the milk or the formation of curd from the milk, which is nothing but the lactobacillus. Okay, now in the milk we have a sugar called as lactose. Okay, so usually what happens is this lactobacillus is responsible for converting your sugar particles, okay, means the lactose based as well as certain proteins which are present in the milk into what? Coagulated form. So coagulated form means what? Coagulated form means clump like thing. Okay, so whatever milk proteins will be there, that will be converted by this lactobacillus into what? A curd form. So here either the lactic acid bacteria, that is one type, or you can take this species which is the lactobacillus. So usually when it is done in industries, what is done is a standardized milk is first, what do you say, it is bought to the industry. Now one important thing over here is whenever the milk is being carried through the containers to that specified factory, okay, for the further processing like making of curd, paneer or whatever milk products are there, the temperature of that tanker should be 5 degrees. So when the milk lot is brought to that particular place, they check the temperature of the milk. Otherwise what will happen is it may curdle, right? So we don't want that to happen. So a standardized milk of a particular fat and SNF has been taken, then it is homogenized. Now what is the homogenized process? Homogenized means there will be different what do you say, sizes of the fat globules in the milk. Now as you can see, sometimes when you boil the milk there will be one oil content floating over the milk. So that is nothing but the fat content. Fat globules will be present. So this fat globules are usually in ununiform size. Okay, so you have to make it in the uniform size. So you do this homogenization, which is basically cutting or agitation of the fat globules into uniform size. Once it is done, you go for the pasteurization process. So pasteurization is what? Just heating the milk up to a different temperature. Like for example, nowadays in the market we also get the tetra packs, right? So it has a shelf life of 90 days, but the same thing, if you take a packet milk, it has what, 1 to 2 days maximum it can be there, right? Afterwards it curdles, or it just, you cannot use this that thing. So for that purpose only you do the pasteurization. So whatever microorganisms which are hazardous to the human beings, they will be killed. Okay, so there are three types of pasteurization temperature. In this, you just have to know what is pasteurization. That's it. Next is what? The cooling. So once it's heated, you have to cool it and then add your starter culture. Now, what is the starter culture? Or it's also called as what? Inoculum. Okay, so what is the inoculum? Inoculum is nothing but basically the lactobacillus culture or the previous formed curd, a little bit sample, you have to keep it back. That is your starter culture or the inoculum. Okay, so as I told you, this lactic acid bacteria, what it does is it produces acid, right? And it coagulates that particular milk proteins. Means it partially digests that particular milk protein. So that function is done by this starter culture. Once the curdling happens, next what you do is the two categories. That is, one is the packing, incubation, cooling, and storing that particular thing. This is the set yogurt if you want. And if it's some other product, then these many things you have to do again. Again, certain incubation, cooling, stirring, and all those stuff is done. So this two flowcharts which you can see down, right? This is basically upon what type of product you're going to make. If it's a simple curd, you can just stop in this particular thing. Okay, but if it's a yogurt which is very smooth in consistency, then you have to go to the second flowchart which is there. So depending upon what kind of product you want, the further whatever the procedures are there, it will be either added or deleted from there. So the major benefit of making curd industrially is whatever the nutritional quality is there, that is increased. Basically, the vitamin B12. Okay, so this vitamin B12 is increased. Now we know that there are something which is called as probiotics. So probiotics means what? Something, the natural microorganisms which you take inside through the bottle, which will increase your good flora of your gut or the stomach, right? So that is basically the role of this vitamin B12. Okay, so what it will do is it will increase or improve your RBCs as well as DNA. Okay, so this is all about what, how actually the curdling of the milk takes place, or how a curd is being formed by using an inoculum culture. So now here if you see, these are what the equipments which you use basically in an industry so that you can what, produce yogurt or the curd. Okay. Okay. Then the next one which we have is what? Now this was one product which is curd, right? The other product which we have is the fermented products. Fermented products. Okay. So now before going into the how the product has been made by using fermentation, you should know what is fermentation, right? So fermentation means what? Whenever a product is formed by in the absence of oxygen, okay, that process is called as fermentation, where a little amount of alcohol is being produced. For example, if you take bread, okay, so in bread we do the fermentation process, and at last when the dough rises, what happens is you will get little bit of alcohol. You won't find it when you taste it, but it gets evaporated when you bake that particular bread. Okay, so fermentation is not limited to a particular product. Okay, if whatever your idli dosa batter is there, dokla batter is there, bread making is there, or you can take alcoholic beverages such as wine or beer and all those stuff, right? So all these comes under the category of what? Fermented products. Okay, so fermentation, in that basically what happens is a certain kind of now there are two categories. Okay, one is either you can say alcoholic fermentation, and the second category is what? Lactic acid fermentation. So if you take the first one, which is the alcoholic fermentation, in this what happens is, now if you take maida, that is the refined wheat flour, or if you take fruits, that is to form any alcoholic product, it has one similar content which is called as what? Sugar. Means glucose is present over there. So basically what happens is the glucose is being converted into alcohol, that is in the form of what? Ethanol. Ethanol. Okay, with the liberation of little bit of CO2 and the energy, that is ATP. So whatever the glucose molecules are there, they have been converted into what? Two molecules of ethanol, two molecules of carbon dioxide with the liberation of the ATP. Okay, so this is what in case of the alcoholic fermentation. But if it is a lactic acid fermentation, what will be the difference? The same glucose molecules only, but instead of the ethanol, what will be produced? Lactic acid will be produced plus ATP. Okay, so this is what the basic, what is known as what? Fermentation. Okay, so through this process only fermented products are formed. So you have different kinds of fermented products. So here the first one which you can see is using yeast in baking. So this is basically to produce your snacks item, like which includes what? The bun, khari, or bread, whatever things are there, right? So how that has been produced is you just take your refined flour, add yeast, sugar, water, form a dough. Now the yeast which is there, it's a type of fungus, right? So this particular yeast, that is the Saccharomyces cerevisiae. That is the species which we take over here in the form of yeast, basically, which you have in the house. So yeast, what it does is it takes whatever the thing I told you, right, about the fermentation. So it just carries out those particular thing, that is conversion of glucose into the alcohol and so and so, and the carbon dioxide. That is why when you add yeast to a particular product, it gets what? Raised up. Okay, whether it's the, now in the idli dosa batter you won't add it because already the microorganisms are present in it, so it does the thing. But apart from that, if you want to speed up your process, you can add the yeast. Okay, so what happens is whatever the sugar is there, that yeast takes the sugar and converts into CO2, your dough rises. Okay, so this is one product, that is the bakery products which you have normally. Then this is the second product, okay, which is the toddy. Okay, so toddy is basically made from the sap of the palm plant. Okay, so as you can see, this is the plant over here, right? The palm plant. From there, you take a fresh palm sap, okay, and naturally you allow it for the fermentation. So two types you are getting, that is the tal toddy as well as the kejur toddy. So it depends upon which palm tree you are taking, a basic palm plant or a date plant. Okay, so this is the second product, that is the toddy. Then, yeah, next which we have is what? The cheese production. Okay, the other important industrial scale, or the product which is consumed by us in a huge amount, which is the cheese, right? So usually when you prepare this cheese industrially, it takes about 20 to 21 steps for a cheese to be produced by using something which is called as what? Rennet, a very important enzyme without which the cheese cannot be formed. Okay, that is what? Rennet. So the basic, or the more amount of varieties of cheese which we use is what? The Swiss as well as the Roquefort cheese. Okay, now what is the difference between this highly demanded Swiss and the Roquefort cheese? So Swiss cheese, actually what happens is, the how you can categorize different types of cheeses depending upon their color, how ripe the cheese is when you taste it, okay, the smell, as well as how much bigger holes are present in the cheese. Now, mozzarella cheese or the simple cheese which you buy, that is of Amul or any other brand for the sandwiches and this, that is totally different. And these two cheeses are totally different. Okay, because as compared to the normal cheese, this will be what? More priced, highly priced cheese these are. So Swiss cheese or the Roquefort cheese, the main difference is what? In the Swiss cheese, you will find that the holes in the cheese will be little bigger. Okay, so whole is bigger means what? It is due to the production of what? CO2. Okay, so there is an important enzyme that is rennet, okay, which is used up by the bacteria, which is Propionibacterium shermani. I'll just write it over here. Propionibacterium shermani. This particular strain is being used to produce Swiss cheese. In the same way, the Roquefort cheese, which is there, that is classified depending upon what? The ripening process. Okay, so how much ripened the cheese is? And that is the type of what? Fungi. And the name of the fungi which is used to produce this Roquefort cheese is what? Penicillium Roquefort. Penicillium Roquefort. Okay, so these are what? The two categories of the cheese and what makes them different and which are the strains used in making these two cheese. So this is a basic step in from which you can categorize how a particular cheese is being made industrially. So the raw milk is taken, pasteurized, standardization. So here you can see the rennet enzyme is there, fermentation, coagulation, that is the curdling is happening, and then the further procedure. Okay, that is when you make paneer also, you get two things, right? One is a coagulated milk, that is the lumps of the milk, and the other one is what? The whey or the watery substance. So same way in the while making the cheese also, you will find the same thing. And depending upon, as I told you, what type of microorganism you're going to use to make the cheese, you will get that particular type of cheese. Okay. So some cheese they actually taste better when they are kept for aging process. Yeah. Now this is the products. These are the products what which are fermented. So as you can see over here, different microorganisms they will produce what? Different acids. That is, they are called as what? Organic acids. Organic acids. Now, organic acids means what? Like for example, in the fermentation, I told you, yeast, they have the capacity to produce alcohol and carbon dioxide. In the same way, if you take acetobacter, they are going to produce what? Acetic acid, right? Lactic acid bacteria means lactic acid. Propionobacterium, propionic acid. So different microorganisms when you take, whether it's a bacteria or a fungi, they have the ability to produce what? Different kinds of organic acids. Okay, which are also called as bioactive molecules. So in this, we also have what? Citric acid. Now, citric acid not only it's present in the citrus fruits, but industrially by using an organism which is Aspergillus niger, you can industrially make what? The citric acid. Okay, so these are what the few examples which bacteria can be used to produce which type of acid. Okay, in this you can also include that alcohol producers, which is the Saccharomyces cerevisiae, which I have told you earlier. Okay, so this is the example of your beverages, that is the alcoholic beverages. So main category which you have is the wine and the beer. Okay, and the basic difference of making this is, if you see this thing, you have what? The fruit juices, right? From which you can make wine, and then the other one which we have is what? The barley. Okay, so usually the difference between the beer and the wine is what? The alcohol content, how much percent it is having. Okay, so compared to the wine, other products which are there, right? The rum, whiskey, or all those stuff which is there, right? So they will be having what? More alcohol as compared to this beer or the wine. Okay, so these are also fermented beverages only. So if you want to make wine, you can use any kind of fruits, especially the grapes are being used, into which the yeast, that is Saccharomyces cerevisiae, is been added, and then wine is produced after several processes. And again, the taste of the wine increases according to the aging. So how many years you're going to preserve it, according to that, it tastes better. Next is what about the beer. So this is usually made from the malted barley. Okay, malted means it's finely a little bit roasted, whatever the barley grains are there, and then they are allowed to sprout. Okay, so when the sprouts come, you just remove it by drying the whatever the sprouts are there. Like physically, you use a sieve to remove the sprouts from that malted or sprouted barley, and then you add yeast and whatever other things are there, and make what beer. So in the beer, there is one important substance or a thing added, which is called as what, hops. So hops are nothing but a thing which is taken from the plants. Okay, so when you add this hops, what happens is it tastes better. Whatever the beer content, alcohol which is there, right? So more, what do you say, smooth consistency beer you will be getting, a foaming consistency you will be getting over there. So this is what, depending upon what kind of material you're going to use and what kind of bacteria you're going to use, you get what, different products. So because if you use barley, you're going to get beer. But if you want this chongu or the rice wine, which we say, right? That if you want to make, you have to use what, rice. So these categories again depend upon whether they are distillated or whether they are made without distillation. Okay, so without the distillation, beer and wine are being made, whereas with the distillation, you have the other categories like the whiskey, rum, gin, and all those stuff. Okay, so this is all about what, the fermented beverages. Now, the next important thing which we have is the penicillin. Okay, so we all know what is penicillin, what are the role it plays even today, right? Regarding our health issues. So penicillin usually was the first antibiotic which was accidentally invented by Alexander Fleming. So antibiotics means what? Usually the chemical substances which is used, means it's basically produced by whom, the microorganism itself, so that it can inhibit what, the other microorganisms which are growing or surrounding it. Okay, so whatever the living competition is there, that has been reduced. So for that, antibiotics like substances are being produced by whom, the microorganism itself. So in the 20th century, this man, that is the Alexander Fleming, he accidentally invented this antibiotic, okay, which is nothing but the penicillin, from the organism, that is penicillium or penicillin notatum. Penicillin notatum. So he found out that this particular chemical which was produced inhibited the staphylococci. Staphylococci. Okay, so when this penicillin was invented, there were two more other person, okay, who further did some research, and they found out that this particular penicillin is having the full potential to cure certain diseases. That's why it was extensively used in the World War II to treat the soldiers. Okay, so even they were awarded what, Nobel Prize for the same in 1945. So the main benefit or the impact of this accidental invention was what, the treatments which were given for your whooping cough, plague, leprosy, those kind of diseases, right, that were cured. And even today, for treating our infections, okay, antibiotics are being used in a what do you say, in more amount, right? So without the antibiotics, the infections cannot be cured, whether you're going for a surgery or whether it's just a simple cold cough. Antibiotics are very important, but the journey of this antibiotic started from here, that is the accidental invention of penicillin. So this is actually in the form of injections or the capsules. But other than that, one more thing which we have is the vaccine. Okay, now here, regarding the antibiotics, I'll show you actually how it looks. So if you see this diagram, okay, now these are small papers which already have the antibiotics. So this was placed in the petri plate which was having the growth of microorganism. So you can see wherever that particular antibiotic the strips are being placed, right, the rounds are being placed there, you can see there is an inhibition, there is no growth of that particular microorganisms. So this proves what, that something is there which is inhibiting the growth of the microorganisms, and you can use it further also. The same way is regarding your vaccines as well. Okay, so whatever the vaccines are there, they also play the same role almost, but vaccines are nothing but the live organisms which you give to the diseases like cholera, tuberculosis, right? So those kind of things, which was invented by Edward Jenner first, and he gave the vaccine for the smallpox. Okay, so that is how the vaccination process also came into the existence. Okay, so this is all about what, the antibiotics. So this is just in the diagram, you can see like how various organic acids, as I told you earlier, right? So what products it gives. Here you can see two more, that is the pectinase and lipase. Okay, so usually whatever the pectinase and lipase is there, they are the enzyme produced acids or substances. So lipase which is there, they, you can use them in the detergent so that the oily stains can be removed from the clothes. Whereas the pectinase which are there, they are being used to clarify the juice bottles. Okay, now usually when we prepare fruit juices, that is of any sweet lime or whatever, you get that particular turgidity in your bottles, right? But it is not the case in the market bottles which you have. They are very clear, just you get the flavor, that's it. So that's because they add this something, the enzyme produced thing which is called as what, pectinase. Okay, so when you take this pectinase and add it into your fruit juice, it has been clarified, means you get clear fruit juices out of it. Then next important thing is regarding the medicinal uses. Okay, so medicinal uses, major three things you have over here. Okay, that is the formation of clot buster, immunosuppressive agents, and the blood cholesterol lowering agents. Okay, so if you take the heart patients or the patients who are having heart attack, they have been given, okay, a thing which is called as streptokinase injections or streptokinase containing tablets. So this, what it does is it acts as a clot buster, means it, a blood thinner it acts as. Okay, so it is given usually for the heart attack patients or the patients who have stents in their heart, right? For those patients you give this streptokinase which has been produced by the bacterium itself, that is the streptococcus. So it's a blood thinner actually. Next you have is what, immunosuppressive agents. So immunosuppressive agents means the cyclosporin A. Okay, so cyclosporin A is being produced by a fungus which is trichoderma, and immunosuppressive means what, whenever the organ transplantation has been done. So there are possible chances of what, that organ can produce infection in that particular organism where this transplantation has occurred in that particular person, right? So to avoid that, you give what, immunosuppressive, because something which enters into your body, which is a foreign particle, your body won't accept it as it is, right? Some kind of reactions, it will try to reject that particular thing, obviously. So in order to avoid that rejection only, you give this immunosuppressive agents, which is the cyclosporin A. And lastly, what you have is the cholesterol lowering agent. Okay, so blood cholesterol lowering agent means statins, which is like the people who are having higher cholesterol and they may have a chance of heart blockage. For them, this yeast produced substance, which is the statins, they are being given. So what it basically does is it lowers your blood cholesterol, means whatever cholesterol synthesizing enzyme is there, that has been inhibited by whom? The statins. Okay, and hence whatever the cholesterol is there, that has been reduced in the body, preventing all other diseases such as the heart attack or the heart blockage.